everyone. I'm Jishnu. I'm an assistant professor at the Center for Systems Immunology at the University of Pittsburgh. I run a computational systems immunology lab. The two founding principles are we think about multi-omic datasets, high-dimensional multi-omic datasets in the context of immune regulation and dysregulation, and we use predictive and interpretable machine learning approaches to analyze and integrate these datasets and do what we think is interpretable machine learning, and I'll talk more about that beyond just predictive machine learning. We also think a lot about biological networks and structures of biological networks, how we analyze and integrate these high-dimensional multi-omic data sets. We actually had 3 talks at ISMB. I'm the last one, so there were 2 of my students presenting talks. However, I'll dive right into slide. So we all know, and there have been many, many deep learning talks at this conference, at both this session and at others, deep learning has absolutely revolutionized prediction. But prediction or getting to correlates or biomarkers is not the only goal. We need to think both about inference, about the mechanisms underlying the data generation and what insights it gives us, as well as prediction. So in human systems immunology, it's not enough to have markers or correlates alone. That's just the what. We also need the how and the why. So to do this, we came up with this latent factor method, and this was a product of quite a long line of work, but this is kind of the flagship method from the lab culminating in all of this. So this was co-led by Javad and Hanshi, 2 awesome graduate students in my group. Javad is the student who asked the last question, and this is also not possible without experimental data sets and my awesome colleagues and collaborators, Alok, Bob, and Amanda. This is something we published earlier this year in Nature Methods. Now, the foundational idea behind slide is very simple. Whatever we can observe are the X's. Those are the features. We want to link these observable features to the response or outcome of interest. That's why, that's standard regression. Everyone in this room knows that. But what really we care about are these unobserved or hidden or latent states. So latent factors and latent models have been around in the statistical literature for 2 decades or more. So we're not saying anything new there, but the latent factor model and the properties that we are using come with very unique properties relative to prior work. The biological rationale behind this is biological methods, techniques that you have at the current point in time, single-cell RNA, single-cell attack, proteomic, metabolomic, they don't really reflect true biological hidden states driving outcomes. They are simply a reflection of technology in 2024. Biology doesn't care about where technology is. We need to parse out these latent states, and then rather than regressing the X to Y, we first discover the latent factors, and then we regress these latent states to Y. So the first point in slide is we have a latent factor discovery pipeline with identifiability guarantee. So this is a standard setup. We're decomposing what we observe into the latent factors and the allocation matrix. But this comes with some very unique properties. The first is, unlike non-negative matrix factorization methods, which are non-identifiable, our method is identifiable. Now, to assume that it is identifiable, there are methods like principal component regression, which make very, very restrictive assumptions like orthogonality of the latent factors and make it identifiable. We can prove identifiability of the latent factors without assuming orthogonality. So these are, unlike NMF, these are identifiable in parameter space, unique latent factors that we get. We solve this as a constrained optimization problem. This was work done in conjunction with theoretical statisticians at Cornell. The only thing we assume is each latent factor is anchored by 2 features. So when we are decomposing the covariance matrix, each latent factor is anchored by 2 features that we call pure variables that belong only to that latent factor and to no other latent factors. Everything else can be shared or not shared in a data-dependent fashion. And so we are allowing for overlapping latent factors because that reflects biological reality. And we are making absolutely no assumptions regarding underlying data-generating mechanisms, so it can be applied to single-cell or bulk data. Now that we have these latent factors, we use a two-step regression. First, we do a linear regression, and then using hierarchical OR logic, we look at the significant terms from the linear regression, and we add on nonlinear terms. But one key facet here is that the latent factors themselves can be overlapping or correlated in terms of genes that load on to these factors. So rather than using the regression coefficients for false discovery rate control, we use something called knockoffs, which some of you may be familiar with, developed by Emmanuel Candice and colleagues for overall false discovery rate control. So we first use knockoffs to identify what we call the significant standalone latent factors. Then using hierarchical OR logic, we look at pairwise interaction terms, use knockoffs again to get significant interaction terms. So compared to many other latent factor methods and many other benchmark methods in the fields, including MOFA, MOFA+, variational autoencoders, principal components regression, partial least squares, we have a combination of all of these properties that other methods don't have. So now we applied it to a whole host of data sets, and in the interest of time, I'll quickly go over two. The first was a data set of scleroderma disease severity, where we had single-cell RNA-seq biopsies, and the endpoint was a surrogate clinical marker of disease severity, the modified Rodnan score. So we took the RNA-seq data, did the usual pre-processing analysis, and asked the question that in a rigorous K-fold cross-validation framework with permutation testing, how well can you infer the basis of scleroderma pathogenesis and disease severity? So the two methods on the left that we show you slide in essential regression is a former iteration of the method developed by our lab, performs as well as or better than existing methods in predicting disease severity. But to us, prediction is the low bar. Great, we can predict. But what actually do we infer? And that's reflected in these latent factors. So each box is a latent factor. It's not based on a pathway, it's not based on a gene set, because pathways, gene sets are context dependent and reflect static priors. This is a context-specific co-expression module that we infer from the data itself. So there's a lot of stuff on this slide. It's a busy slide. I'll walk through it very quickly. The ones with the green boundaries are the significant standalone latent factors. The ones with the pink boundaries are the interaction terms. The color represents a cell type of origin for the transcript, and things on the left of the dotted line are associated with higher disease severity, things on the right associated with lower disease severity. The more important thing is, what did we find mechanistically? We recapitulated known mechanisms of crosstalk between myeloid inflammatory signaling, interferon signaling, activated fibroblasts, myofibroblasts that are well known in the field to contribute to pathogenesis. Without using any prior knowledge, slide recovered these mechanisms. But we also found new things involving keratinocytes. So people mostly focus on fibroblasts and myeloid cells, but we found an intriguing circuit involving crosstalk between fibroblasts, myeloid cells, and keratinocytes, as well as a T-cell latent factor that we validated in downstream experiments, which I'm not going into here. More importantly, the effect sizes that we find outperform what other latent factor approaches, including MOFA+ and VAE, find, and we really identify both linear and nonlinear relationships. We also asked a question: if we are really inferring something meaningful, then in a perturbation experiment, we should be able to recover known mechanisms of action. So this was a drug, tofacitinib. Basically, this is a pan-JAK inhibitor, and we looked at patients before and after treatment, and we said, well, if this is really helping us infer something meaningful, does it recapitulate the known mechanism of action? And the only major latent factor that we see is reflective of IL-6 JAK-STAT signaling. This is, of course, an organismal phenotype. We also applied it to a cellular phenotype. This is paired single-cell RNA-seq data and TCR data from a non-obese diabetes model of type 1 diabetes, where cells were labeled based on their extent of clonal expansion: unexpanded, low expansion, moderate to high. We are again able to predict clonal expansion as well as or better than other state-of-the-art methods. But more importantly, we are able to infer these context-specific groups, which include naive CD4 T-cell markers, activation markers, and inhibitory receptors. Some of these are well-known molecules like LAG3, PD-1, well-known activation markers, inhibitory receptors, as well as markers of naive CD4 T-cells like L-selectin and CCR7. But what really matters is the group structure, and the group structure overall outperforms existing latent factor approaches picks up things that univariate DE analysis would miss. So only some of the genes in the latent factors would meet univariate significance, others would not. And the context-specific group is what's important. And you can see this here. If you look at well-known markers like LAG3, TIGIT, PD1, CD200, they are higher in the expanded, cell CCR7 higher in the unexpanded. But when we look at the groups, they recapitulate this structure. We have applied it on many different data sets, which I don't have time to talk about here, including spatial data sets, because this makes absolutely no assumptions regarding underlying data generating mechanisms. So in summary, SLIDE can integrate high-dimensional multi-omic data sets with no assumptions regarding data generating mechanisms to identify significant latent factors. Our putative causal and some inferred mechanisms have been validated using perturbation systems or model organisms, comes with rigorous statistical guarantees regarding identifiability and corresponding inference, FDR control, and the discovery of nonlinear hierarchical relationships. And we've identified putative factors in a range of phenotypes from organismal to cellular and molecular. And this is my lab. We're a lab of around 25 people looking at different things, all related to the immune system. And I would like to thank my funding sources. None of this would be possible without my funding sources. Thank you so much for the invitation.
Thanks for the talk. We have time for some questions. I will start with one. So you emphasized a few times the context specificity is important, right? So how do you incorporate the context, and what does it mean actually in your case, the context?
Yeah, so that's a great question. When I say context-specific latent factors, what I mean is we identify the Z matrix without any biological prior. So we are using, let's say you have single-cell RNA-seq data, spatial transcriptomic data, whatever, single-cell attack data, we are identifying the structure of the latent factors solely based on the data itself. So these groups of genes that we see in the latent factors do not rely on any prior gene set or pathway definition. It's inferred from the covariant structure of the specific context of interest.
Maybe another question is, how do you annotate your factors? Is that done afterwards manually, or is that done automatically?
That's a fantastic question. So there are two ways we think about it. One is you could do some basic stuff like doing overrepresentation analysis or gene set enrichment analysis and see which pathways they relate to. They are sometimes informative, but sometimes not, because the key claim is that this context-specific group may or may not match prior biological gene sets and pathways. So what we've done is, in addition to these basic analyses, we've also overlaid it with, let's say, inferred gene regulatory networks, and we've done in silico perturbation experiments. And we see that it overlaps with what you would expect biologically in terms of GRN structure, let's say, but it only captures part of it. So it's really giving you the putative driver inferred from the data.
What was the difference between the green and the red genes in your boxes?
One associated with higher disease severity, one with lower. Yeah.
All right. Thank you so much.